going to talk under the topic cellular respiration and fermentation, which we are going to divide it into three big parts, which are aerobic respiration, anaerobic respiration, and the differences between them. Okay, so for aerobic respiration, it's a process that breaks down food by using oxygen. The reactant for aerobic respiration are glucose and oxygen, and the product from aerobic respiration are carbon dioxide, water, and energy. ATP is how energy is stored. So aerobic respiration has four stages, which are glycolysis, preparatory reaction, Krebs cycle, and electron transport chain. So first, we'll take a look at glycolysis. So glycolysis happens in the cytoplasm and is divided into two stages, which are investing and harvesting stage. Okay, let's take a look at the process. The process. First is investing phase, and it is 2 ATPs required to rearrange glucose by attaching 2 phosphate to 6 carbon compound. The 6 carbon compound will split up into 2-3 carbon compound with each phosphate. Then harvesting phase. The 2-3 carbon compound then lose electron. The 2 NAD plus will gain that electron and convert it to NADH. Then it goes to electron transport chain and back, carrying electron and phosphate. The phosphate will attach themselves to 3 carbon compound, giving 4 phosphate. Then the 4 phosphate donate itself to ADP and converting itself to ATP. After that, the 3 carbon compound change itself to another 3 carbon compound called pyruvate. Okay, now for input and output. The input is glucose, 2 ATP, 2 NAD+, 4 ATP, ADP plus P. And for output, it's 2 pyruvate, 2 ADP, 2 NADH, and 4 ATP. For preparatory reaction, this is the second step in cellular respiration. It occurs in the matrix in mitochondria. It's also known as pyruvate oxidation. And the picture down here is showing how preparatory reaction works. In the process of preparatory reaction, there are 3 main parts, which are first, 2 pyruvate from glycolysis will enter the mitochondria to process preparatory reaction. Second, 2 pyruvate is oxidized to 2-acetyl-CoA. 2 NAD plus will reduce to 2 NADH, and 2 carbon dioxide will release out of the mitochondria and out of the body. Then last, 2-acetyl-CoA will attach to coenzyme A. And for the input and output of preparatory reaction, the input are 2 pyruvates and 2 NAD+. The output are 2 acetyl-CoA, 2 NADH, and 2 carbon dioxide. After preparatory reaction is Krebs cycle. The Krebs cycle is the third step in cellular respiration. It occurs in the matrix. It also known as citric acid cycle. For the process of Krebs cycle, it will start at 2-acetyl-CoA from preparatory reaction will enter Krebs cycle one at a time. So in order to break down 1 glucose completely, we need 2 rounds of Krebs cycle. The acetyl-CoA will attach to 4 carbon compound, forming 6 carbon compound. Then 6 carbon compound is oxidized to 5 carbon compound, and NAD+ will reduce to to NADH, and carbon dioxide will release. Next, 5 carbon compound will oxidize again to 4 carbon compound, and NAD plus is reduced to NADH. Carbon dioxide is releasing. And then 4 carbon compound will arrange itself and oxidize ADP to ATP. Then 4 carbon compound will rearrange itself again, transferring its 2 hydrogen atom to FAD, and FAD will become FADH2. Lastly, 4 carbon compound is oxidized and NAD plus is reduced to NADH, and 4 carbon compound will be reused in the cycle again. To sum up, the input for Krebs cycle are 2 acetyl-CoA, 6 NAD plus, 2 FAD, and 2 ADP. Output for Krebs cycle are 2 4 carbon compounds, 6 NADH, 2 FADH2, 2 ATP, and 4 carbon dioxide.
Electron transport chain is an aerobic respiration. Electron transport chain is the last step of the cellular respiration. It is the only part that uses oxygen as an electron acceptor. Electrons are passed from the point to the last point. It is happening in the crystal membrane. It produces the most ATP. The oxygen combines with the hydrogen ion to produce H2O or water. So basically, electron transport chain is the last step of the cellular respiration. It makes the most ATP. So the process are, the first thing is NADH got oxidized to NAD plus as it lost electron from the hydrogen to the electron transport chain. Then the hydrogen, when the hydrogen lost its electron, is there a proton left, so it changed to H plus or hydrogen ion. Number 2, FADH2 also got oxidized to FAD, and it also lost electron to the electron transport chain. Then when they lost electron, the high energy electron moved through the chain as it releases the energy where it is moving. The energy that electron release are used to pump H plus to crystal of the mitochondria. It creates a concentration gradient from the low to high concentration or active transport. So the active transport are when you move from low to high concentrations gradient. When the low energy electron reach the end of the electron transport chain, it will leave and go to the oxygen, which the last electron acceptor. So when the electrons are moving, it lost its energy. So when arrive there, the energy is low. And oxygen carries the hydrogen ion to form water or H2O. H plus moves down the concentration gradient into matrix by ATP synthesis. Then it helps ATP synthesis make ATP by adding a phosphate to it. Input and output of the electron transport chain. The input are NADH and FADH2. The output are 34 to 36 ATP. So as I say, the electron transport chain produces the most ATP.
In anaerobic respiration, glucose breaks down without oxygen. The chemical reaction transfers energy from glucose to the cell. Anaerobic respiration produces lactic acid rather than carbon dioxide and water. Unfortunately, this can lead to painful muscle. The process of it, glycolysis, is the first stage of respiration, in which the glucose molecule is broken down into 2 pyruvate molecules, releasing electrons in the process and generating 2 molecules of ATP. Instead, pyruvate is converted into different byproducts through a fermentation process, and carbon dioxide is released as well. 2 ATP molecules are generated in the process, and the output is 2 lactate or 2 alcohol and 2 carbon dioxide and 2 ATP net gain. And the types of it. There are many types of fermentation that are distinguished by the end products, form of pyruvate or its derivatives. The 2 fermentation most commonly used by humans to produce conventional foods are ethanol, Fermentation and lactic acid fermentation. Alcoholic fermentation. Alcoholic fermentation is the basis for the manufacturing of alcohol beverages such as wine and beer. Lactic acid fermentation. It makes ATP in the absence of oxygen by converting glucose to lactic acid. The advantages and disadvantages. The advantages, it provides a quick burst of ATP energy for muscular activity, but the disadvantages is lactate and alcohol are toxic to the cell, and lactate change pH cause muscle fatigue. For the differences between cellular respiration and fermentation, aerobic respiration will require oxygen. On the other hand, anaerobic respiration does not require oxygen. The reason why aerobic respiration requires energy because in the electron transport chain step, if oxygen is not exist, the proton cannot be pumped to intermembrane space and the concentration gradient cannot be created. The result from that is we cannot produce ATP. Next, aerobic respiration occurs in mitochondria and cytoplasm. But anaerobic respiration occurs only in cytoplasm because anaerobic respiration will mostly occur in microorganisms and they don't have mitochondria. And aerobic respiration will produce more ATP than anaerobic respiration. Aerobic respiration will mostly be found in plants and animals, such as human and tree. But anaerobic respiration will mostly found in microorganisms, such as amoeba and fungi. In addition, anaerobic respiration can also be found in marine organisms because in the deep sea there are not much oxygen there. As you watch this video until now, this is the end of our video. Thank you for watching and listening.